To introduce Rebecca Bart to give today's seminar. She did her undergrad degree at Reed College and then did a PhD in plant pathology at UC Davis, which is where I met her and interacted a bit. And then she went on from there to do a postdoc with Brian Staskowitz, where she started working on Cassava. And then now she's been in her own position at the Donald Danforth Center for the last 4 years. So
we're modeled after an academics department, so I'm an assistant faculty member and hopefully soon will become an associate faculty member. I do have graduate students. I'm an adjunct professor at Washington University, and I also have postdocs and technicians in my lab, and every once in a while I'm able to lure a naive undergrad from WashU to come and work with us. So that's what we are, and that's where we are. And I thought I would just start today. I realize this is a plant pathology department, but I wanted to just make sure we're all speaking the same language. I think one of the things that I've reflected on throughout the day and the conversations I've had so far is that this department is a really nice spectrum of all the way from extension and really applied up to the very nitty-gritty molecular and little bubbles on PowerPoint slides, as I just experienced with Justin. So let's just make sure we're speaking the same language. And also I wanted to remind everybody, or tell you for the first time if you don't know, a little bit about the history of how we think of this concept called the gene-for-gene model of resistance. Of course, this started with Gregor Mendel and just the observations that he made about inheritance. That was later picked up by Robert Koch in the germ theory, and this was really proving that microbes are what cause disease, and it's not some other weird thing going on. Here I'm choosing to acknowledge one of the many people who contributed to the discovery of DNA. Of course, that was very transformative in our understanding of what I'm eventually going to get to. And then the gene-for-gene resistance model was really originally coined by Henry Flor in the 1940s or so. And I also want to pick up Barbara McClintock, because her work was really revolutionary in terms of really understanding the genetic structure of how DNA works. So here's my highlight of heroes. And what all of this is coming to is this concept that we know of as the gene-for-gene model of disease resistance. And all that simply says is that there's a gene in the host and a gene in the pathogen, and both of those have to be present in order to get resistance. And a lot of this original work was done before we really knew about how proteins interacted with DNA and how type 3 secretion systems were injecting proteins into plant cells. But over the last several decades, the system has gotten much more complicated, and I'm sorry this is all blurry. I checked my slides right before this talk. But it's okay that this is all blurry because I'm not going to talk about any of this. And the only reason I show this slide is to tell you I'm not going to talk about any of this, but to emphasize just very quickly how complex. It started from this very simple observation of gene-for-gene interaction. And over the last many decades, through a lot of really hard work, the molecular aspect of this, very complicated signaling networks have been discovered to describe the resistance that plants display. We've also been able to observe on a molecular level how plants become susceptible and how pathogens can actually use mechanisms to proactively create that susceptibility. And those are the two, on the host side of my talk, those are the two different dichotomies that I'm going to talk about. I'm going to talk about an active resistance response on the side of the plant, and an active susceptibility process on the side of the plant. Just to highlight a little bit of foreshadowing, I'm going to be talking about bacterial pathogens. Some of the susceptibility targets that we and others have studied are these sweet genes, and I'm also going to be talking about some viral pathogens in interaction with some host proteins. But I promised I wasn't going to talk about all of those really complicated signaling cascades. And so for the purposes of my talk, I'm going to distill it down even further. And I'm going to use a series of cartoons here that are very simplistic, overly simplistic, but hopefully instructive for the talk. And if we think about plant innate immunity in this simplistic context, we can distill it down to 3 stages, where we have basal resistance. This is where a pathogen might be, or a prospective pathogen might be recognized by the plant cell pattern recognition receptors and triggering low level of resistance. In the actual context of what we see in a plant, we don't really see much. A successful pathogen, for example, the Xanthomonas pathogens that I've spent most of my career working on, have developed some way of blocking that basal immunity as well as inducing susceptibility. And I'll be talking a lot about this water-soaked symptom here, so I try to give you a blow-up of what that looks like. And then the final step here is when the plant has developed the ability of recognizing, I'd either directly or indirectly that pathogen and triggering a strong resistance response. And this is often accompanied by a cell death phenotype. Now, my lab looking forward, I'll wrap up with some just sort of musings about where we're going. And that's this environmental side of the disease triangle. We know that the environment influences disease. I'm sure everybody in this room understands that. But the reason I wanted to start my talk today with the sort of remembering of Mendel and Flor and all of the different pioneers who led to our understanding of the gene for gene resistance is what I spend a lot of time thinking about right now, is how can we as scientists develop that same sort of concrete mechanistic understanding of how the environment is influencing these interactions. We know it does, but we don't have a really good model that's broadly applicable. So keep that in mind as I go through the talk. So we're already done with the intro. The rest of my talk is going to be broken up into at least 2 and possibly 3 if I have time topics. First, I'm going to tell you, I tried to just kind of get a grab bag of stuff that hopefully something will be interesting to everybody in the room. The first one is really a sort of classic plant pathology story of a re-emerging disease of cotton that my lab started working on. After that, I'm going to really shift gears and talk about some of the crop improvement efforts that we're working on in Cassava. And then finally, if I have time, I'll expand a little bit more on this idea of the environment's role in plant pathology. Okay, so new mystery. This is cotton. Probably most of the people in the room are wearing cotton, so I probably don't need to spend a lot of time defending it as an important crop. This is the disease. This is cotton bacterial blight. It creates these necrotic lesions that start out as water-soaking spots. It's a vascular Xanthomonas pathogen. It was one of the major limiting diseases of cotton in the early and mid-1900s, and then breeders did what breeders do very well. They identified a bunch of resistance genes, moved them into germplasm, and the disease was controlled. In, I think it was 2013 or so that I first saw this on Twitter, but this was one of the first Twitter posts that alerted me to this new emerging disease. This is Tom Allen. He's an extension specialist at Mississippi State University, and he's a highly trained plant pathologist, was wandering around in a farmer's field and saw this disease that he had learned about in textbooks, but had never seen with his own eyes. It's been fun. I've collaborated pretty closely with Tom since that initial discovery, and on the work I'm going to tell you about. It's been fun to sort of follow his moods as, this disease has spread. So here's more recent from this year, 2017, where he said several calls regarding fungicides being applied to manage cotton bacterial blight. Fungicides don't work on bacteria. Twitter's funny. So when I was first trying to learn about this disease, I checked Wikipedia, of course. There wasn't a whole lot on Wikipedia, so I did actually go to a library. And this was a really fun process, just to really dig back into some of the old literature and read about the early breeding that was being done in the 40s and 50s to combat this disease. There was also a more recent review, an annual review of phytopathology from 2005, that really summarized a lot of this earlier work. But needless to say, there wasn't really a lot of recent stuff published on this disease. What I'm showing you here is just what we have observed over the last several years for this disease. This was 2011 when it was first recognized, confined to just a couple of states. And then you can see fairly clearly it's spreading as time goes on. You'll notice I don't have a map of 2013, and we still don't really know what happened that year. There just wasn't a lot of reports of it. Perhaps the environment was less conducive to the disease that year, or perhaps people were just busy with other stuff and it didn't get reported. We're still really not sure about that. To overlay where we're seeing the disease with cotton production in blue and the disease outlined in red, you can see that it's spread to many of the most prominent cotton growing regions. And if I say cassava ever when I'm talking about cotton, I apologize. I do that all the time. This whole first part of my talk is cotton, not cassava. Okay, so my graduate student, let me just acknowledge her quickly. This is Ann Phillips. She's a graduate student in my lab, my very first graduate student ever. And she really was intrigued by this and has spent the last several years trying to figure out what's going on. So she had a number of hypotheses when we first started this project. Her first hypothesis was that perhaps a new species or strain of the pathogen was introduced into the United States. As a plant pathologist, this was kind of exciting. Maybe it was a new species that made a host jump. Maybe it was a new race of the pathogen that had been introduced somewhere else. And maybe we could take a genomics approach and actually look at the evolution of the pathogen and answer that question. The different species of Xanthomonas. And this blue circle right here is the newly isolated strains that I had received from Mississippi, as well as a couple that we had been able to get from a culture collection that were from the 80s and 90s when the disease was just kind of piddling out. We then wanted, so this basically told us that it wasn't a new species. It wasn't some species from tomato that had done a host jump and now could cause disease on cotton. We next did a little bit more of a fine-tooth phylogeny to see if we could see, well, maybe it's still evolution of the pathogen away from those historical strains. And that's what I'm showing you here. So this is a phylogeny based on full genome sequencing of all of these different isolates. And over here is race 18. This is the clade of the pathogen that was most common early on in the 40s and 50s. And here you can see race 18. This is one of those historical strains. And then several of the different strains here and here that I had collected in Mississippi. So it's really just looking pretty much the same. No major phylogenetic shift. So that hypothesis was wrong. Next we were thinking, well, okay, so no major phylogenetic shift, but it could still be a subtle mutation that allowed the pathogen to overcome that resistance. So the way that we decided to look at this, here I'm showing you my cartoon again just to remind you that resistance proteins interact either directly or indirectly with effector proteins. And so perhaps there had been some mutation in one of these effector proteins. So the way we looked at that is we just looked at the effector proteins in all of these different genomes. What I'm showing you here is a bunch of different strains of the pathogen. Here are all the strains down in that race 18 clade. And then here are a bunch of the type 3 effectors that are in those pathogens. There was 24 conserved effectors that were present in all of the different strains, and then a bunch of other ones that were variable, but within our race 18 clade were all the same. So next we did the thing that was a little bit more obvious to do, which was to simply go and take a bunch of cotton varieties that we knew used to be resistant or used to be susceptible and re-inoculate them and say, are the new strains still triggering those reactions? And the answer was simply yes. The ones that historically have been resistant are still resistant. The ones that were historically susceptible are still susceptible. So our final hypothesis was the one that was a little more emotionally draining, which was that farmers are simply planting susceptible varieties. And so what Anne did here is she took two different data sets and collated them. We have a collaborator in Texas that has been screening all of the most common cotton varieties for resistance and susceptibility forever. And so we took that, and then we took a data set that is available to the USDA about how much acreage is planted with each of these different varieties. And what Anne saw was a very clear, disturbing trend of the number of susceptible varieties, or the percentage of susceptible varieties planted each year going up, 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 up. So unfortunately, that means that it wasn't any of the really exciting hypotheses that I had introduced earlier. The good news is that it means that there is still resistance out there that's effective against this pathogen. And so this made us just wonder a little bit about, well, why? Why did the breeders in these breeding programs stop selecting for this resistance? Why were they releasing the susceptible varieties? And I think if you think about it for a minute, it's pretty obvious. Every time you try to select for another trait, that's money, that's the bottom line. And we also know that a lot of these resistance genes are genetically linked to things that are not so great. So there probably was a fitness benefit. In the absence of the pathogen, a fitness benefit for getting rid of it. So now the question is, what can we do about it? Well, we have identified sources of resistance in germplasm that are still effective against this pathogen. This is an ongoing collaboration with Basu Kuraparthi at NC State. He's been developing some really extensive resources for cotton. Here's a phylogeny of, I think, 384 or something cotton varieties that he has been studying. He sent us 52 varieties from a NAM population that he's developing, and we screened those and identified at least 9 different sources of resistance that are phylogenetically distant on this tree and therefore are most likely independent sources of resistance. So we can exploit that. We also took a look at the other side of how plants respond to pathogens, the susceptibility aspect. Work from my group and others have shown that a lot of these Xanthomonas pathogens use tall effectors to which are a specific class of bacterial protein that are injected into the plant cell, travel to the nucleus, and induce things like sugar transporters to export sugar out into the apoplast. So we did an RNA-seq study on this pathosystem and found, very similar to other pathosystems, that there are in fact halofactors, and they do in fact induce sugar transporters. We also found another potential susceptibility target, which if anything I think might actually be a better target in this pathosystem, which was a homolog of the mildew locus O, which is a fairly well-known resistance gene that has previously been published to confer resistance to various different types of pathogens. So to summarize that, we have identified the contributing factors to disease reemergence. We got to do some fun genomics and molecular biology, even though at the end of the day, the story was a little bit less exciting. So the good news here was that we identified several promising strategies to control the disease. I can also say that the companies are now paying my collaborator, Terry Wheeler, in Texas to screen their varieties again, so that's good. The bad news is that in the course of this, we kept hearing calls from people, especially in Texas, saying that they had planted resistant varieties, but they were still showing symptoms of this blight. And I really just thought that, oh, they mixed up their seeds, or I kind of ignored those calls for a while, until eventually we received samples and actually did Koch's postulates ourselves and found out that there were in fact 2 different pathogens in those leaves. And so what we now know is that there's a new cotton pathogen. It's a Pseudomonas. This is the Xanthomonas here that we've been studying, and here was this mysterious white bacteria that came with it. And very troublingly, showed here, when you inoculate the 2 pathogens together on cotton that's resistant to the Xanthomonas, we get this sort of spreading necrosis phenotype. I have another picture of it here. So here's the Xanthomonas by itself causing that water soaking or a hypersensitive response on the resistant varieties. Here's the Pseudomonas, what we call now Pseudomonas syringae malvacearum. And on the susceptible cotton, we get a lot of spreading necrosis shown here, but even on the resistance, we get a fair amount as well. And so moving forward, we're really interested from a more basic side of plant pathology. We're just really interested in trying to understand how these 2 different pathogens are interacting. There's several mysteries here that we don't fully understand, one of which is that although we've shown pretty clearly here that the Pseudomonas can cause disease on its own, we've never been able to identify it in the field on its own. It always is tagging along with the Xanthomonas. So that might just mean that we haven't looked hard enough, or it might mean that it really is a symbiotic relationship between the 2. And so we have a couple of different models about what that might look like. In a plant cell that is susceptible to Xanthomonas, you might imagine that these 2 different pathogens are complementing each other and turning on different susceptibility pathways that are mutually beneficial. And then I think the more interesting side of the equation is what's happening over here, where we have a resistance protein, presumably recognizing that Xanthomonas, and that that's being somehow turned off either upstream or downstream of that signaling by the Pseudomonas. And just to wrap up, I had a nice talk with Gwen this afternoon about sort of how people like us are going about learning about new diseases. And I think this is a place, I'm just learning about them on Twitter. There's another Xanthomonas corn disease that I've been following more recently. But there's got to be a better way, so perhaps some of you can chime in if you want. Before I move on to my next story, I'm going to just stop quickly and thank the people who did this work, especially Ann Phillips, my graduate student. She's funded by NSF. Jeff Berry, Jill Berg, Anupama Vijayaraman, Mark Wilson, Mariah Norman, and other undergrads also contributed to this, as well as our collaborators, especially Tom Allen, who was originally the one who identified if anybody wants to chime in with a question while I take a drink. But it's okay if you don't. Yeah, go ahead.
Is there any information yet about how the Pseudomonas might be spread in the environment?
Nope. Hopefully soon. Hopefully soon. Yeah. What?
Did you take another host that's susceptible to one of the Pseudomonas syringae pathovirus as a foliar pathogen and to Xanthomonas and see if you could repeat that?
So if I'm understanding your question, asking whether or not the Pseudomonas might be broadly able to help xanthomonads?
Yeah.
So like, for
Okay,
example,
so
in
the-
cassava?
Yeah, since there's certain effectors that cause water to be released in Arabidopsis, if you had an xanthomonas that went in there, could you find the benefit?
Yeah, that's an interesting question. I think I did suggest that to one of my postdocs who's working on sorghum, because he's working on a xanthomonas pathogen of sorghum, and he's been seeing some Pseudomonas in some of his isolations. But I said, well, try and Pseudomonas as well. I'm not sure if he tried it yet. I guess I would sort of expect that it would trigger a hypersensitive response because it does have its own suite of type 3 effectors, but it might not. So we should give it a try. Did you have another question?
I guess so. Have you counted colonies? Like, do you see more colonies of Pseudomonas in the presence of Xanthomonas or vice versa?
Mm-hmm.
I just think of viral synergy or something like that.
Yeah, so that's work that's actively ongoing right now. Anne is looking at that both just in culture, if you grow them together in culture, either minimal media or rich media, and then also isolating them back out of the plant. But I don't know the answer yet. But I think it's a good thing to look at, and I'll hopefully at the end show you some of the other kind of tools that we have to look at that. Okay, well, why don't I'm going to move on. I'm really going to change gears now, so just prepare yourself for a mental shift a little bit. The other thing that my lab has been working on very extensively for a while now is just trying general crop improvement efforts in cassava. So before I get into what we're doing, let me just tell you all a little bit about cassava. How many people feel like they know a lot about cassava? Have you eaten cassava? Yeah. It's good, yeah? I think it's good. Okay. So for everybody else, this is cassava. This is what you find on Wikipedia. Here's a picture I took in Nigeria of cassava sort of grown under ideal conditions. These storage roots are massive. They're like the size of my trunk. It does produce flowers and seeds pods in the field, but not in the greenhouse, and so that's been a challenge to work with it. Here's kind of it. Somebody was asking me earlier about where cassava is grown. Here's sort of a distribution of that. It's processed in a lot of different ways. A lot of people eat it just kind of cut up and boiled or fried. In Western Africa, it's often processed, as well as in Asia. A lot of different uses. But one of the things that I really, ever since I started working on cassava, I've just been wondering is why. Why is it is this, I believe now, firmly, and there's a lot of data to support that it is one of the most important crops in the world for its role in food security. But why is it so popular? Why are people eating it? Here's a little bit of a heat map that I put together, where over here on the left, I've listed all of the different things that we like to find in our food, vitamins, minerals, proteins, and then each column is a different crop. We've got maize, rice, wheat, potato, cassava. And so what you can see here is cassava doesn't have a whole lot of most of the things that we like to find in our food. It's not particularly good for you. To make things worse, a lot of varieties of cassava actually have high levels of cyanide in them, so if you don't process it correctly, it can make you very sick. There are sweet varieties of cassava that don't have that problem, but most of the ones that are grown in the countries I work on do have this problem. They also suffer from this thing called post-harvest physiological deterioration, which basically means that when cassava is still underground, it is very stable. It can stay there for a long time until a farmer needs it. Once it's harvested, you have about 24 hours before it turns, 24 to 48 hours before it turns into a big pile of mush. So it basically has no shelf life. So why? Why cassava? Well, it is this amazing food security crop, partly because of how pest tolerant it is. It is generally grown without fertilizer, without irrigation, and it thrives quite well. It is very drought tolerant and can grow in poor soils. Here's a picture I took in Tanzania of a cassava field that I just actually stumbled upon. I was on a walk. I thought I was kind of in an inhabited part of this island I was on, and I looked down and there was a cassava plant thriving. You can see it's basically in a pile of rubble here. So, you know, it's pretty amazing. It's a pretty amazing crop, right? I also like to, before I get into the research I'm doing, point out that most of our modern day crops, for example, corn, that I realize a lot of you know about, this represents from teosinte to modern day corn, about 10,000 years of pretty intense domestication, right? A lot of the work that I've done in collaboration with some people at JGI, resequencing most of the farmer-preferred varieties of cassava across sub-Saharan Africa, have shown us that when we compare them to the wild relative, they're about 3 or 4 generations from their wild relative. So thinking about this timeline and then how good cassava is already, I just think is a point to reflect on a little bit. The final reason is if you work in cassava, you might get to meet Bill Gates. But cassava does have some challenges. As I've already mentioned, it does flower, but not easily in the greenhouse. Its genome is not that bad. It is a diploid organism. Its genome is about 760 megabases, so not, as plant genomes go, not too bad. It does have a long generation time. The variable flowering phenotypes make breeding really difficult too. You have to synchronize the flowering. Some varieties don't flower very well at all, others flower profusely. There's also a high degree of heterozygosity. There are no inbreds in cassava. There is no hybrid vigor, so the productivity that we're seeing is sort of, you could think of it as natural hybrid vigor, sort of. I mean, it just never went through that process to become inbred and then have targeted process to make the inbreds. We also do have a reference genome, but it's very fragmented. There are at least 3 major diseases of cassava in sub-Saharan Africa. South America has a couple of more diseases, but I'm going to just tell you quickly about the 3 that I'm working on right now. Here's Cassava bacterial blight. Well, here's a healthy cassava field in Uganda. This is what happens when the pathogen goes through. This is a vascular pathogen, and so in the worst, most extreme cases, you get this real defoliation and death of the crop. There's a couple of different viral diseases. This is a Gemini virus that causes cassava mosaic disease. Here's a picture of the symptoms on the leaves. This is a really fascinating story that I'm not going to have time to talk about today. I will talk about this one though, which is cassava brown streak disease. This is a potivirus, and here we're showing you some of the symptoms in the leaves, and let me just emphasize that these are really, really extreme symptoms. Most of the time, this disease in the field doesn't necessarily produce very much foliar symptoms. The real symptoms are when the farmer harvests the crop and sees that this has happened to the storage roots, and more importantly, these brown necrotic lesions have formed. And we really don't know much about the physiology that's leading to this, but it makes these roots completely inedible, complete loss. So one of the things that I've tried to do, so I started my talk with the cotton and talking about learning about emerging diseases from Twitter and how that's a resource that we have here. One of the things that I'm continually asked with these diseases of cassava is, well, how important are they? How many farmers are affected? How much loss is there? Where are they most important? And that information has been really, really difficult to get. Partly, not because people aren't collecting the information, but because there's teams of researchers employed through the national programs across sub-Saharan Africa that go out to the field and they take really good data, very important data, and then put them in books on shelves. So one thing that I've tried to do is simply just digitalize that information so that we can look at it. I went to Uganda last summer and sat around in a room for a very painful 2 days with my collaborators, Titus Alakai and his team. And we were able to actually get a lot of that data into notebooks and start to look at it. So here I'm just showing you 2008 to 2013. We can see in the blue line is that mosaic disease. This is the only cassava disease that has strong genetic resistance. And so that downward trend simply shows that it's working and that farmers are adopting the varieties that have it. Cassava bacterial blight in red is very variable. This is really classic of a bacterial disease. Some years it's really bad, some years it's not so bad. And then in green is the cassava brown streak disease. This one doesn't really have any natural genetic resistance that's been identified, and it's definitely increasing. And perhaps even more troubling, it's really, right now it's constrained to East Africa, but it's moving west. But we do have a really robust transformation pipeline, and so this opens the doors for applying biotechnology to disease control. I want to just quickly tell you about our transformation pipeline. We start with leaf explants, bring them into tissue culture, induce organized embryogenic structures, which we call OES. These get turned into friable embryogenic structures, which is the material that we treat with agrobacterium, and then plants can be regenerated. So in my lab, since I started my lab, we've really tried to jump on the boat for genome editing as a possible technology to generate novel resistance strategies for cassava. So let me just tell you quickly, probably most of you have heard about this, but if not, I really recommend learning about it because it really is revolutionizing pretty much every field of biology right now. The way it works is there's a protein Cas9 that complexes with a guide RNA. The guide RNA can direct this nuclease to a specific place in the genome and induce cutting. If you just induce cutting and leave it at that, you can get mutations as the host repairs that cut. It's an imperfect process. You can also introduce a repair template with homology and the sequence that you wish was in that location. And at some very, 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 very low frequency, you get homologous recombination and swap in your sequence, your desired sequence. So I'm going to summarize in one slide about 3 years of work. This was funded through the Bill and Melinda Gates Foundation in collaboration with Dan Voitas and Nigel Taylor, where we were really just trying to develop gene editing technologies for cassava. And in this project, we started out focusing on herbicide tolerance, not necessarily because we want to deploy herbicide-tolerant cassava across the world. I think there's a lot of issues with that, potentially, that I'm not going to get into today, but I'm happy to discuss. But the main reason we targeted this is because this is a trait that we can select in culture. We don't have to get plants out of culture and into soil and grown up to test them with viruses to see if we've been successful. And this turned out to be really critical in terms of very quickly developing this technology. And in the end, we were able to not only do just targeted mutation with genome editing, but we were able to do that very rare thing that I mentioned, homologous recombination, where we supplied a 4.2 kb repair template, and we're able to replace the promoter all the way through the second exon of the EPS-PS gene. And when we treated those plants with glyphosate compared to the wild-type plants, we saw a really nice phenotype. Since then, we've done a fair amount of tinkering, trying to do other things. We messed with some of the flowering components and got really, really pathetic little plants that tried to flower in culture. We also did some work with PDS that I'll come back to. But primarily, I want to talk to you about this one more applied strategy for the cassava brown streak disease. So this collaboration is with Jim Carrington and Brian Saskowitz at East Berkeley, and stems from early work from Jim's lab with Arabidopsis, where his lab and others discovered that there's these EIF4E or ISO4E mutations that confer resistance to another type of podovirus. And the way this works is through the interaction with the VPG of the viral RNA. And so we thought, well, maybe we could apply a similar strategy in cassava, but the first question was, which of these many different components should we target? We took two different approaches to try to narrow this down. We looked phylogenetically at all of the different homologs in cassava, shown here with the man's IDs, and there was 5 different potential homologs in cassava that seemed like good candidates. And so Michael Gomez, a PhD student that I supervised when I was still in Brian's lab and continue to collaborate, conducted a yeast-2 hybrid assay with all of those different homologs, and then with the VPG domain of 2 different strains of the virus. And he saw 2 obvious hits that popped up. But this brought up the question of, well, maybe we're going to have to hit both of them in order to get a phenotype. So at this point, my postdoc, Dan Lin, kind of took over the project and generated the constructs and regenerated mutations. And what he did was he knocked out both of those genes independently and then also targeted them both simultaneously. And so what I'm showing you here is some emotionally not particularly satisfying phenotypes. I'll be clear about that right now. It gets better. Hold on. Here's wild-type plants, uninoculated, and then the same wild-type plants showing symptoms. And what we get is sort of these brown, dark brown necrotic lesions on the stem, as well as the sclerosis on the leaves. If we knock out just one or the other of these novel cat binding proteins, which are EIF4E homologs for all intents and purposes, we don't really get a very strong phenotype. You still see the lesions. When we knocked out both of the novel cat binding proteins together, we have 2 different independent lines. The leaf phenotype was still there, but we seem to have a reduction in the stem phenotype. And so we decided, well, okay, we need to repeat this a lot and see if it's really robust. It was definitely not as strong of resistance as we were expecting. Other papers, when you mess with this interaction, really saw strong immunity. So Dan did a lot of different experiments. I'm just showing you one here. He did this many times with multiple different strains. Let me just tell you quickly how these experiments are done. So here we have average incidence. What this is Cassava is propagated clonally, so we can take one event and then replicate it many times. And then we can track how many, over time, how many plants are showing any symptoms. So this plot has nothing to do with symptom severity, it's just how many plants, if you have 10, how many of them are showing any symptoms. And when you see the lines going up and down here, that's because Cassava is growing and it sheds leaves as it grows. So what we saw was the wild type and both of the single mutants very quickly had pretty much 100% incidence of the virus. And then both of the double mutants showed significant reduction. He also looked at an average disease score. So this is sort of a combined average of both the leaf and the stem phenotypes. This data was a little bit more messy, but there was still an obvious difference between the doubles. And then he did area under the disease progression curve. And obviously this was statistically significant. What was perhaps more striking and impressive was when we dug those plants up and looked at the actual roots, because as I acknowledged early on, the symptoms in the leaves are not always so strong with this pathogen. And here we felt a little bit better. Here's wild type showing the necrosis in the roots, and then one of our double mutant lines, pretty clean. Dan also tried to look at virus titer, and again, I'd like to highlight that this was obviously not immunity. The virus was still there, even though it was significantly less than the wild type. So we were really scratching our heads a little bit about why we hadn't gotten as strong of a phenotype as we had been hoping for, and also what else might we be able to do to get a better phenotype. It's one of the things that's really difficult about working, especially on something like Cassava, having partial immunity and then deploying this thing. There's so many risks with doing that as well, because if it's not going to show a good phenotype and you put it out there in the field, you're going to lose trust. We take those situations very seriously. So let me just tell you, this is a little bit more speculative of what we think is going on. I'm going to tell you about it, and you all can, especially the virologists in the lab or in the room, can shoot me down. So here's the gene model of our two different novel cat binding proteins, and here's where the guide RNA was targeting. And in retrospect, this was really early days when we designed these constructs. In retrospect, they were really way too close to the intron-exon boundary, and that made our lives more complicated than it needed to be. That decision was based on the location of a restriction site, which would have made it very easy to tell whether or not we had gotten mutations. But either way, we did get mutations, and those are kind of cartooned down here. I know it's a little hard to see, but here's the wild type. The capital letters are the exon, and then it moves into the intron. And so for our double mutants, we see a couple of different mutation phenotypes that we got for line 2 and line 8. Here we got a relatively larger deletion, here just a single nucleotide in this gene, and then over here too. And so what Dan started thinking was, well, maybe by targeting these mutations at the intron-exon boundary, we haven't actually disrupted the protein sequence. And so he did cDNA-seq, as he calls it, where he just amplified the cDNA and PCR cloned that from different E. coli colonies. And what he saw was for wild type, here's again the exon, intron. In the wild type, we see the wild type reading frame in 7 out of 7 clones. For the novel cat binding protein double mutants, the two different lines, we saw what we call the type 1 splicing coming out. So this is the one that's most, I guess, alarming, because what we think is happening, what we call type 2 splicing, is that we're splicing right here for exon 2. And so you get this internal extension, which adds amino acids to the protein, but doesn't actually shift the reading frame. What we don't know is whether or not this type of splicing also occurs in the wild type, but is just not the dominant isoform. And so I know that's a little bit hard to follow. Some of you are really frowning at me right now. Some of you are nodding. So let me just try to give you my take-home message here, which is that I think it's important to recognize that most of the genes that we think about in plant genomes or any genomes, are defined by a dominant isoform. But if that dominant isoform becomes non-functional, alternative isoforms might be able to compensate, and that we just need to be aware of that when we're designing these strategies for targeting different genes. A final twist on this that I want to just introduce with genome editing is I think it's really important to conceptualize your tissue culture pipeline. The other thing that we have seen a fair amount is that it's possible to get chimeric plants arising. And the reason this happens is because you start with a plant, you produce your FECs, you treat it with agrobacterium. There's nothing to say that you don't get multiple cells transformed. In fact, you hope you get multiple cells transformed. Those cells will start dividing and replicating. At some point, your genome editing constructs are going to do the job that they're supposed to do, but they might do them at different times, and this can then lead to chimeric plants being regenerated. And just recently, I have published a paper with a Ugandan PhD student that I co-supervised, where he just looked at this in a little bit more depth in cassava by targeting that PDS gene. So I'll refer you to the paper if you have interest. Okay. So before I go to my last little topic, I'm going to subject you, hopefully not too much against your will, to a movie. And the reason I'm doing this is, before I play it, let me just introduce. Ever since I started my lab, I've been really interested in this idea that the environment affects, as I've already told you, affects the disease severity that we see, but we don't really have the tools necessarily to get at the mechanism. If you want to get at the mechanism, you have to have really carefully defined systems. That's something that I think we can all kind of appreciate. And so I certainly don't have the answer yet, but one thing that my lab has been doing is just trying to be a little bit more creative about the different tools that we can use. And about a year ago, I needed to take a little break from science. Something happened. And that's not my movie. So I put together this movie, and it's just kind of fun, and it tells you about some of the tools that we're using. So hopefully it will play for you and you can enjoy it. So one of the things that we use is this high throughput phenotyper. One of the problems we have is just with replication. And yes, it is set to Daft Punk. I can turn it up. And so this tool gives us the ability to just get really highly replicated experiments. It holds about 1,000 plants, and we can put them through and image the plants every day, so we can do automatic watering. We can choose to water these ones a little bit and these ones a lot. But what's more powerful is what's shown here, which is what we call the imaging loop, as soon as it turns, of the plants going through. And so this gives us RGB images every day, as well as NIR and fluorescence, though the fluorescence camera hasn't really been particularly useful. And so we're able to ask things just like, how do the plants grow? We've been using this more recently for some of the microbiome work that we're doing. NIR has really been fairly helpful, especially for the disease resistant stuff, where we know that these pathogens can change the water content in the plants. Almost going on to the next tool. The plants have to go home. So there's some of the statistics on it. The other thing that we've been using, I don't surf, my husband does, but it's this bioluminescent marker. And so we've been using this to try to watch how the pathogen, this is just a pretty movie of circadian rhythm, it has nothing to do with my research, but it's the same technology that we use to be able to watch the pathogen moving through the plants. It's going to come in a minute, sorry. Yeah. So there's the bacteria, we've expressed Lux in it, and what this movie is going to show you is the pathogen moving through the plant. And so this has really been a fun tool. I mean, it's just fun, we get to watch bacteria colonize plants, but we can also do things like moving the plants to different humidity chambers and seeing how does that affect. So not only are we able to look at disease severity from the disease point of view, but we're able to watch it from the bacterial point of view. So that's my pathogen. Here's a viral pathogen that we study. It took me a long time to get the little leaves to flap like that. And then the final thing, those are kind of more expensive, high price ticket items. We've also been using Raspberry Pi imaging chambers a lot. So here is an example of just watching cassava grow and die. It turns out that I'm going to stop it there. We also do lots of engagement with the community. Anyway, the whole movie is available on YouTube if you want to just enjoy it over and over again. So let me just, in the last couple of minutes, reflect on what I just told you. We are really interested in the disease triangle. This is a picture that I would never, ever, ever have asked undergraduates in my lab to take, but when they chose on their own volition to take it, I was very happy. They each had a project that encompassed a different side of the disease triangle. And so what we're really trying to do is we're trying to move from these sort of traditional disease assays, not move away from them because they have a place. They're really statistically robust and manipulatable, but we're trying to interface those now with some other sort of low cost and high cost options. And I'm just going to play one more movie for you really quickly because it makes a point that I think is important. So this is the same movie that was in the video of the Cassava, it just goes a little slower. And so, you know, I always wondered that picture I showed you early on of the cassava field in Uganda and the defoliation, why does that happen? And why does that happen so quickly? So by just setting up a little Raspberry Pi camera and watching the cassava plants grow, these two did not have the pathogen, these ones did. We were able to figure out that it actually happens very, very quickly. You're going to see this in a minute with this one where it's trying so hard. It's really still struggling. And then in the course of about 3 hours, it dies. And it's so sad. But that just really, really told us about how quickly, and now we know with the bioluminescence, of course, that what's happening there is that the pathogen is clogging up the vasculature, and it's as though you've taken away the water to the plant. So hopefully, going forward, we're going to be able to use some of these more creative, sensitive tools to really probe at this central question of the role of the environment in disease severity. So with that, I would like to just pause for a moment before I take any questions and thank the people in my lab. Ann Phillips is the one who did all of the work with cotton. Dan Lin has been leading the work with CBSD. And then it's a very collaborative lab, so everybody has worked very hard together. And this is what we look like most of the time. And then also really thankful to a number of different collaborators, internationally and nationally. And then specifically the funding. Cotton Incorporated has been a really big supporter of the cotton work and is supported by an NSF fellowship, and I have a little bit of money from the USDA from that. The Cassava Brown Streak work was supported both by the Two Blades Foundation and the Bill and Melinda Gates Foundation, and then work on sorghum that I didn't have time to talk about from the DOE. So with that, I'd be happy to take any questions. Wrapping up exactly.
Questions?
What kind of work is being done on the post-harvest deterioration?
Great question. There's at least some amount of work just sort of taking a transcriptomics approach to try to understand what's going on there. There was a couple of recent papers, but I don't think there's a mechanism that's really solidified yet.
Do you feel like there's any reason why you can't make it as stable as potato?
I don't think that there is any reason. I think it just hasn't been done yet, and that's where I really feel like Cassava has this untapped potential where we can, it's exciting because we should be able to really relatively easily apply knowledge from other systems to Cassava. It definitely has its own, like, let's see, Nigel Taylor, my colleague, always says, it'll beep you every time. I don't want to swear in front of all of you anyway. It always throws some unexpected thing at you, which is really annoying, but anyway. Yeah.
So, I just thought of something and that's your next question. How difficult is it to make genetic resources like populations and then do bulk segregation analysis on, like this post-harvest trait, so you could localize some area that might be causing it and then maybe edit that afterwards. So is it a problem to make, if it doesn't flower in the greenhouse, can you make crosses in the field? Is that useful, or is it one of those organisms that's kind of, I don't know anything about cassava.
Yeah, so it is useful. We have the project, one of the projects I didn't talk about for the Gemini virus, there is classical resistance for that, and we've been trying to clone the gene. We were able to make crosses in the field and do some genetics on the F1 population, especially because the parents in that case are so heterozygous, they're basically like your F1 anyway. So it is possible. I think the bigger constraint that we face with cassava is that most of the genetic variation that's currently in most of the farmer preferred varieties across sub-Saharan Africa come from this one breeding program, the Amani breeding program that happened in the 30s, and it's like one cross with a wild relative. And then it's really just been a couple of additional rounds of breeding and then distributed. And so what I would like to see happen, and what I think there's no reason it can't happen, is to go to those culture collections in Cali, Colombia, and to take a more global approach to some of these questions. It's very possible that they already have a variety that they've developed that doesn't suffer as much from post-harvest deterioration, and that just by using sort of GWAS approaches, we'd be able to get at some of the underlying mechanisms. But the communication across the different communities that are working in Cassava is not what it should be yet. Yes?
The first presentation on Cotton, you talked about Pseudomonas and
Yeah.
is there a possibility of like this complex interaction, like between the two particles, like the synergistic interaction?
I think so. I think it is a synergistic interaction. We haven't yet discovered the mechanism, especially because in the lab, we can take the Pseudomonas and get it to cause disease on its own. And so we're still really interested in trying to understand why do we see it in complex with the Xanthomonas? Does it just happen to be there and they're kind of ignoring each other, or is it actually more of an active engagement? I would guess that it's more of an active engagement based on what we've done so far.
Any other questions? Thank you.